Hi there! Welcome to Cornell iGEM's Molecular Bio Crash Course. My name is Nicole, and today I'll walk you through some slides. A bacteria is a single-celled organism and they come in multiple types, but we usually work with a bacteria that looks like this. It's sort of this rod shape with a plasma membrane. Bacteria is definitely not as complex as a human cell, but they do have some similarities. The similarities of interest for us are DNA, ribosomes, and plasma membrane. We can take advantage of DNA, ribosomes, and plasma membranes to give the bacteria characteristics that are favorable for a project. You'll notice that the bacteria has a cell wall and a capsule. These two features help the bacteria survive, but if we're trying to change the bacteria's DNA, we need to get past the cell wall and capsule, and I'll explain this in more detail. And next slide. So after you find a model organism, now you can get to the genetic engineering part. When we talk about genetically engineering a bacteria, we don't mean that we're changing the DNA within the bacteria itself. Instead, researchers found an extra chromosomal piece of DNA called plasmid. Extra chromosomal in the sense that it's not dependent on the bacterial DNA to replicate or maintain itself. A plasmid is basically an extra piece of DNA that can replicate itself and usually gives bacteria beneficial traits. You may have heard that antibiotic resistance is rising, and plasmids can give bacteria antibiotic resistance, among other things. What makes plasmids so great for research is that we can cut them with something called restriction enzymes, which are specific enzymes that can cut a plasmid at specific locations on DNA, called a restriction site. For example, there is a specific restriction enzyme that cuts the DNA in this step-like manner you see in that bottom picture. Before we get to the actual process of designing a plasmid, it's important that you understand the central dogma of molecular biology. This diagram shows that information can only flow in one direction in the same predictable pattern. You'll always have DNA converted to RNA, which is then interpreted by ribosomes to make proteins. It's important to remember that DNA, both the bacterial DNA and the plasmid DNA, can only store information. It can't interact with other cell components like ribosomes or foreign invaders in the way that RNAs and proteins can. However, this is a good thing because this makes DNA a stable place to store information. Therefore, we can mess with the DNA in order to make different RNAs and proteins for our own purposes. Since editing the bacterial DNA is beyond difficult, we engineer plasmids instead. A plasmid can be engineered to have a bunch of different things, but it's important to remember that a gene can only produce RNA or proteins. If your project revolves around producing bioplastics or carbon sources, for example, you'll need to find workarounds. For example, a gene of interest can be an enzyme that can help you make bioplastics. Also, plasmids rely on the incorporation of genetic circuits, which is basically an assembly of biological parts for some function. Each circuit usually has 4 elements. The promoter is basically what tells the bacteria, hey, there's a gene here and we need it to be made into a protein. The RBS, or ribosome binding site, contains a special DNA sequence that says, hey, start converting the gene into an RNA from here. It's labeled as the 5' primer site on the diagram. The gene of interest, labeled as inserted gene on the diagram, is the genetic sequence for whatever protein or enzyme or thing you're trying to make. And lastly, the terminator tells the bacteria, hey, stop making the protein here. Now, after you make the plasmid, you have to get the bacteria past the cell wall and plasma membrane, and we do this with a protocol called a transformation. It's basically a fancy way of saying we throw a plasmid into the bacteria. The protein coding genes in that plasmid will then be expressed since the bacteria has ribosomes. Note that in the smaller picture on the screen, there's a red color. This red is created by RFP or red fluorescence protein. When you design your plasmid, you can include the genetic sequence for red fluorescent protein, or RFP. Therefore, after a transformation, if you see red, then you know that the transformation worked. There's two major types of reporter constructs, which are fluorescent proteins and chromoproteins. All right, that's it for today's crash course. Hope this was helpful.